Okay, let's take a look at molecular genetics. The first part of molecular genetics, you need to know how to differentiate between chromosome, gene, and DNA. So what is a chromosome? In order to understand chromosome, you have to understand that a cell has cytoplasm, have nucleus. Whatever that's inside the nucleus, they are known as chromatin. Chromatin will condense and form chromosome. So basically, chromatin looks something like this, long thread-like structure. Chromosomes would be drawn as something like this. Okay, Ken? Hello? So a gene is a segment of the chromosome. So if it's found in chromatin form, then this small segment is the gene. If it's in chromosome form, then this small segment is a gene. Okay? Ken? Yep. DNA actually is made up of 2 strands. If I were to split them, it's 2 strands. This is DNA. Okay? How to connect chromosomes to DNA? So remember, I mentioned that chromatin condenses into chromosome. The reason why it looks something like this is because the chromosomes have condensed version like this. All the chromatin are coiled up like this. While this one, all the chromatin are coiled up like this. So that's why we draw it as something like this. Short form. Okay? Yes, Harpreet? You get me? So if I were to take one of the chromatin and extend it like this thingy here, I will erase part of it. It is a long structure of chromatin. Chromatin is made out of DNA coiled around protein histone. There is a protein histone over here, which I put it in red. In other words, chromosome is equals to DNA plus protein histones. Okay? Hello?
Yeah.
So if I were to drag DNA like this, and this will be a segment of DNA. This is called gene 1. Maybe gene 2 is smaller. Then gene 3, gene 4. Okay, the location of the gene and the length of the gene is random. Okay?
Yeah.
So basically, chromosomes equals to many genes. So many genes will make up chromosomes. And then a gene is a segment of DNA. So basically, this link chromosome to DNA, and then this link chromosome to gene, and then finally this link gene to DNA. Can?
Yeah.
So I want you to copy this down because they like to ask you to, oh, can you describe the relationship between chromosome, gene, and DNA? So these are how they are linked to each other. They are relationship. Can you copy this down first?
Yeah.
Okay.
What is a histone?
Histone is a protein. Remember the red circles just now? Basically, if I draw the chromatin, I draw a line out of the chromosome. The chromatin, make it bigger and bigger and bigger. It will wind around, DNA will wind around protein histones. This one. These are histones.
Okay.
Histones are positively charged protein because DNA is negatively charged, so it cannot coil around itself because negative and negative will repel. So it needs to bound or wind itself around a positively charged molecule to neutralize itself. Then only it can coil around like this. Okay?
Okay, thank you.
When you're done, you let me know.
Okay, yeah, I'm done.
Okay. So in molecular genetics, you need to learn about the basic unit of DNA. Basic unit of DNA is basically your nucleotide. Just like the basic unit of starch. Basic unit of starch is glucose, the simplest form. Basic unit of protein is amino acid. Basic unit of DNA is nucleotide. Basic unit of human will be cell. Okay? Good? Hello?
Yep.
Abrit, do you follow me?
Yeah.
Okay, so you
Hello?
know you need
Yeah,
to
yeah.
you need to learn how to draw nucleotide. Nucleotide has a phosphate group, has a deoxyribose sugar, and a nitrogenous base. So if question asks you to draw a nucleotide, this is what you should draw. And then remember to label them. This one refers to phosphate group. This one refers to deoxyribose sugar. You want to write pentose sugar also can. Or this one is nitrogenous base. Okay? I want you to take a piece of paper and draw this down because later I'll teach you how to draw DNA. So this question is about draw a nucleotide. Okay, can you draw this first?
Okay, can.
Then next, draw a DNA strand. How to draw DNA strand? You have to draw 3 nucleotides at this side. Can you draw this first?
Okay, can.
And you connect them. This edge over here connect to the phosphate. This edge over here connect to the phosphate. So basically this one refers to one, sorry, this should be DNA molecule. This is one strand of polynucleotide. That means you have one nucleotide, two nucleotides, three nucleotides, many nucleotides together, we call polynucleotide. Okay. Janet, are you done with marking hormones? Janet? Janet?
Yeah.
Do you have the worksheet for the next topic?
What is the next topic?
What's your school doing after hormone?
I think we're doing inheritance.
Doing inheritance. Okay,
Yeah.
so in that case, I want you to go to the chat, and then try and copy down the YouTube link, and then you go and watch. Is that okay? Later
Okay.
I'll get you the worksheets.
Yeah, okay. Thank you.
Harpreet, let's come back to this. So basically the other strand will look something like this. Same nitrogenous base, but it's the other way around because it's anti-parallel. This one is facing up. This one is facing up. This strand will be facing down. You see the pentagon is facing down. This one is facing down. This satellite disk also facing down. Then you connect this. So this is how you draw a DNA molecule. A DNA molecule has two strands of polynucleotide. And you draw at least 3 to show 2 connection. The other side, you also draw 3 connection. And then the 2 strands are connected through the nitrogenous base. Is that clear, Harpreet?
Yeah. Do we connect the nitrogenous base together?
Yes. But to be specific, you have to draw properly. You have ATCG. So one of them will take after this shape, which is a sharp shape. The other one will take after a round shape, semi-circle like this. Take note, one of them is longer than the other. So if this is, oops, if this is A should be the bigger one, T is the smaller one. G is the bigger one, C is the smaller one. This one is sharp. This one is semicircle. So if this is sharp, it will take after this. So A is the bigger one, T is the smaller one. Okay?
Yeah. Okay.
And then A and T has 2 hydrogen bonds. G and C has 3 hydrogen bonds. This one got 2. So this is how you draw a DNA molecule.
What is it? Is there a difference for the hydrogen bond?
Sorry?
Oh, you said the AT is 2 hydrogen bonds and then the GC is
GC is 3
3, is
hydrogen
it?
bonds. This is due to the shape of their nitrogenous rings. So
Oh, okay.
those nitrogenous bases, right, they come together at a certain angle, resulting in the ability to form hydrogen bonds. So G and C happens to be able to come very closely in 3 points. So in that case, it formed 3 hydrogen bonds, while A and T, due to the structures different, it can only form 2 hydrogen bonds.
Okay.
Mm. Okay, so so this is the most of it. Then finally, let's look at the Chargaff rules. In Chargaff's rule, Irving Chargaff, he actually went through the hassle of finding out ATCG. Remember the nucleotides? Oh, the nucleotides that are nitrogenous base, right? There are 4 types of
Yeah.
nitrogenous base, which is ATCG. A for adenine, T for thymine, C for cytosine, G for guanine. Okay, so he went through the trouble of go and analyze, say, rats. He take rats whole DNA, like it go and take the nucleus. Say this is a cell of the rats. These are all the chromosomes. So he go and take out all the DNA and analyze their percentage of A is to T is to C is to G. Although the value is not as nice as this, but what he concludes is he noticed most of the organism that he analyzed has close to 1:1 ratio between A, T, 1:1 ratio between C and G. Herring is a type of fish. Say it has 17%. T will also be close to 17%, probably 17.3 or 16.9, something like that. C would be 33, something like that. He go and start doing analysis on everything, of course human as well, and notice all this. This value is just an example. It's not the accurate value. I'm just throwing out some numbers so that you can understand that they are actually 1:1. Okay. So basically, Chargaff noticed such a pattern. So after he noticed such a pattern, he started to come up with a certain theory. So he mentioned that a lot of organisms will have a ratio of 1:1 for its A is to T and a ratio of 1:1 for its G and C, when the organism has genetic material that is double-stranded. Okay, so double-stranded, for your level, you'll be learning about DNA being double-stranded, RNA being single-stranded. Okay, but it's not entirely true. There are double-stranded RNA in some virus, but that one is not in your syllabus yet. For you, your level, you just learn strictly about DNA being double-stranded, RNA being single-stranded. Okay, so in other words, the genetic material of that organism must be a DNA lah. That's why it's double-stranded. It must be double-stranded, then only you follow this rule. So that is the first prerequisite. The second one, they must follow, the genetic material must follow complementary base pairing. Meaning, A can only pair up with T, C can only pair up with G. Remember the shape that I showed you just now? A that look like this can only pair up with T. A, which looks like this, cannot pair up with A. A also cannot pair up with C. Wrong shape. A also cannot pair up with G because it's too big. Okay, Ken?
Yeah.
Happy?
Yeah, can.
So normally, frequently asked question will be, they will give you a percentage like this. And then say 37% and ask you to fill up the rest. So what you do is, if this is 20, then you fill up 20 lah. This is 37, then this is 37. Then you take 50 minus 20, you're going to get 30, 30. Then you take 50 minus this, you're going to get 13, 13. Something like that. Very easy. And then there's a follow-up question. They will say that, oh, scientists went into the deep sea and then find out, discover a new organism. So this new organism doesn't follow this ratio 1:1. Then they'll ask you, why do you think, can you suggest a reason why this organism doesn't follow Chargaff rule? So the reason would be, very likely this organism, this new organism, it has a genetic material that is single-stranded. So it likely uses RNA as its genetic material. That's why it's single-stranded. That's why it doesn't follow Chargaff. Okay, Ken?
Yeah.
Okay, so one more thing is, I will show you why double-stranded is needed. Say I have single strand. Single strand, normally if you only have one strand, you can't really predict the percentage. So say I have a random amount of A, 1A, 2T, 3C, 4G. All of them are not 1:1, right? Correct?
Yeah.
So if I were to make it double strand and complementarily pair them up, A pair up with T, T pair up with A, C pair up with G, G pair up with C. So what happened here is, earlier you have 1A, 2T. Now you have 1, 2, 3. You have 3A, 3T. So it falls into 1:1. Okay? Ken?
Yeah.
Yes. So
Yep.
earlier you have 3C, 4G. Then now you will have 7C, 7G. So it falls into place. 1 is to 1. So these two are the requirements to basically follow Chargaff rule. Okay, Ken?
Yeah.
Okay.